sorry. Okay, let's start the class. So we were talking about aerobic respiration in the last class, and what we have discussed up till now is we have talked about the first and the second step of aerobic respiration. And what is the first and the second step? It is glycolysis and Krebs cycle. So we have talked about the glycolysis. One important point about, or 2-3 important points about glycolysis is, first, where does it occur? It occurs in the cytoplasm. Second, is it oxygen dependent? No, it's not oxygen dependent. Third, it is a common pathway. Why is it called as common? Because it's seen in case of both aerobic and also in case of anaerobic, right? What is the product that is synthesized? It is only ATP that is synthesized. And if you are talking about ATP synthesis occurs in two pathways, that is via ETS or substrate level phosphorylation. So this is something about glycolysis we have talked about. Next, because of glycolysis key form kore pyruvic acid. Pyruvic acid needs to get broken down further so that carbon dioxide and water gets evolved, and obviously with the evolution of or the synthesis of ATP. And so there is something called as a link reaction, right? Pyruvic acid converts itself to acetyl coenzyme A in the perimitochondrial space with the help of a lot of enzymes, that is an enzyme complex. The reaction is decarboxylation occurs. So ultimately that ki hoy carbon dioxide ulai. Ar jehetu link reaction is also a part of respiration, so ATP production o hoy, kintu substrate level ne via ETS, obviously via ETS, right? Next is glycolysis is linked to Krebs cycle through the link reaction. After the link reaction, acetyl coenzyme A that is produced because of the link reaction enters inside the matrix of mitochondria where the Krebs cycle starts. When acetyl coenzyme A enters inside the matrix, someone needs to accept it also. So we have an acceptor molecule of Krebs cycle. Who is considered as the acceptor molecule? Oxaloacetic acid, right? Oxaloacetic acid 4 carbon, acetyl coenzyme A 2 carbon, 4 plus 2 6, citric acid cycle, citric acid kiman compound 6 compound. And within this cycle we see at certain steps decarboxylation occurs, and ultimately we start the process from oxaloacetic acid itself. So 6 carbon opera 4 carbon hobo lagile decarboxylation kebar hobo laibo. Two times. So we have 2 steps where decarboxylation occurs. Next, what we said is 1 molecule of glucose breaks down into 2 molecules of pyruvic acid. 2 molecules of pyruvic acid gets broken down into 2 molecules of acetyl coenzyme A, and so Krebs cycle needs to turn twice. No, eta molecule glucose completely break down by complete oxidation hobo lagile Krebs cycle kebar solibo laibo dubar. Talking about Krebs cycle or the TCA cycle, we have talked about the different products that are synthesized, right? As far as the reducing cofactor is concerned, NADH2 and FADH2, and also GTP. Together with these energy aru ki produce kore, obviously carbon dioxide, right? So what is the amount, what is the number of carbon dioxide synthesized, what is the number of ATP that is generated, ATP generate hoy ne GTP generate hoy, NADH2 K bar generate hoy, FADH2 K bar generate hoy. This is something that we have already discussed. After that, we were talking about something called as the electron shuttle, right? So what we remember from the electron shuttle, that what is electron shuttle? Nothing but a carrier system, right? What does it carry? It carries the NADH2 from where? From the cytoplasm to the inside of mitochondria. NADH2 cytoplasm keneke ahile because of glycolysis, right? And depending on who carries the electron, either the cell will develop will have 36 ATP or they will have 38 ATP per glucose molecule. Next, today what we will discuss is we will be writing about the third step of aerobic respiration, that is called as terminal oxidation. As far as explanation is concerned, terminal oxidation explanation we have already completed in the last class. Talking about terminal oxidation first, terminal oxidation kiho karne lage, because its most important product water gets synthesized because of terminal oxidation. ATP also gets synthesized because of terminal oxidation. That is the reason why terminal oxidation consists of 2 steps. Eta kami ki boli ko? And that ki for water, right? Aru eta ATP. So phosphorylation what to do thakibo, right? So we have 2 steps. One is called as the ETS and the other is called as oxidative phosphorylation. Talking about ETS or electron transport system, what we said, the electron carriers kisuman thake. Kot thake? In the inner mitochondrial membrane. Atat ki thake, we have talked about the presence of certain types of complex. Within the complex, there are certain type of components. What would the components do? Would take in the electrons and protons. After that, what will happen? Once the electron and protons come out from the matrix to the inner mitochondrial, to the inner membrane, electrons move from one complex to the other and from there to the next. But what about the protons? The protons are pushed out from the inner mitochondrial membrane. NADH to eta molecular para electrons bila jete eta complex or para antu complex hole jabo, proton push out kebar koribo, tinibar. 3 pairs of protons are pushed out, and that is the reason why we see 3 ATP getting generated from one NADH2. If you are talking about FADH2, the electron and protons of FADH2 also goes along that ETS, and protons are pushed out from the inner membrane, but how many times this time? It's just twice. So as a result, what we say, 2 ATP form from one FADH2. Another important point that we have discussed, something about the complex. First, how many complex are there in the inner mitochondrial membrane? 5 complex. Out of this 5 complex, electrons bilake keta complex of kumabo pare? Sarita. What are they? First, second, third and fourth. As far as the 5th complex is concerned, this is the pathway of whom? This is the pathway of the protons. Protons bilak kor pra humabo? Perimitochondrial space or pra akko matrix of humabo. So that 5th complex could also be considered as what? We will have a term called as coupling factor, or we can use a term called as FOF1 particle, right? So hekhini explanation already ho goise, aji ami notes khini likhim, right? Last ki likhisila just amyl oxidation, okay. ekhine loke to sobe buji paisa sake ha team yeah, you are going to get this answer once we'll draw that sorry once we draw the ETS, right? Kuntu keneke jai ki hoy, once we draw that, you'll get the answer. Instead of me saying it right at this moment, first ami nije diagram to akilom, tarpor ami buji pam. Hobo?
Mitochondria code exactly ATP function.
Sorry, code exactly ATP form hoy inside the matrix.
Kintu mane inner membrane
No hoy, inner membrane keneke hobo. Inner membrane through protons bilak humai he jabo. Inner membrane ki ase? F1 particle. F1 particle ase. FO part 2 kot lagi ase. Inner membrane khonot head part 2 towards the matrix protrude hoy goise nai. Bostu to enekwa nohoy jano. Enekwa nohoy jano. So ekhon inner membrane. Inner membrane etu part ase. Etu part ok ami ki kom? FO. Etu part ki? F1. The F1 part 2 kot ase. Matrix. Ekhon ki hoy? Inner membrane. So iar pra jeta protons bilak push koribo because of proton gradient. Push kora pisot ki protons bilak yate roi jabo niki? Yate roi ne jai no? Yale ahi jabo? Kole ahi jabo? Matrix ole ahi jabo. Kintu protons bilake yat first ahibo, tapsot yate jetia protons ahibo, tetia hale ki form koribo? ATP is enzyme. Hey enzyme to a bhitorot thaka ADP tour logot IP join kori pelai ki form korai dibo? ATP. So ATP code form korile. Matrix. No, it's matrix. Answer ki inner membrane dia ase. Tumi question to bhalke dekhaba. Thik ase? Okkoman kiba twist to thakibo pare tate. ATP kotha khudise ne ATP azor kotha khudise? ATP kotha khudise? Thik ase ma question to sam ha. Christie bhitorot nohoy. Christie thake ha. So hey Christie bilak ko thake inner membrane hey Christie, suppose etu inner membrane no, etu Christie, thik ase? Ei Christie jun bilak ebur thake tak ami ki buli koiso? Proton pathway. Ultimately ekhon tomar inner membrane hol, to yato complex thakibo no, etu complex thakibo, ei gutei tuku eta complex buli dhorim. So ultimately Kihor outer membrane? Mitochondria. Mitochondria ekhon hol tomar outer membrane. ইনাৰ মেমব্ৰেইন তো এখন যদি ইনাৰ মেমব্ৰেইন হয় এইবিলাকত ক্ৰিষ্টি থাকিব আমি ডায়াগ্ৰামতহে এটা দেখাইছোঁ ডেট ডাজ নট মিন যে অকল এটাই আছে ডেট ডাজ নট মিন যে অকল এটা এটিপিয়ে প্ৰডিউচ কৰে অনবৰটো এটিপি প্ৰডিউচেই হৈ থাকে ৰাইট sorry kahi ase I forgot to bring water also today sorry লিখিবা নোটসটো sorry কি হৈছে যে এটা পাঠ হল না? এইখিনি পাৰ্ট হৈছে ন Cytochrome B, cytochrome B, cytochrome C and C1, cytochrome A আৰু A3. C1 হয় cytochrome C and cytochrome C1, cytochrome A আৰু cytochrome A3 আৰু cytochrome what is important is you have to remember the sequence ঠিক আছে ইমান মাজৰ পৰা কোৱেশ্চন নাহিলেও লাষ্ট cytochrome যোনে নেকি লাষ্ট ইলেক্ট্ৰন একসেপ্টৰ হ'ব ন তাৰ নামটো খুদে out of in the cytochrome complex which cytochrome is considered to be the last electron acceptor acceptor নহয় হি ultimately donor হৈ যাব হিটো তাৰ নিজৰ তাত ৰাখি নিদিয়ে ন so last is A3 so question has been asked maximum about A3 so A3 আৰু question টো আহিব পাৰে oxygenক electron to কোনে transfer কৰে বা কোনে donate কৰে it is always A3 that will donate the electron to the oxygen right Oh, it's copper containing. ठीक है सर cytochrome A और cytochrome A3 copper containing. Okay, মই কি কৈছো যে cytochrome portionৰ এই sequence-টো মনত ৰাখিব লাগিব যেহেতু electrons বিলাক এটা particular sequence-ত আহিব না complex 1 পৰা complex 2 complex 2 পৰা complex 3 so cytochrome বোৰ তেনেকে specifically arrange আছে এনেকুৱা নহয় যে A3 প্ৰথম থাকিব তাৰপিছত কিবা C থাকিব তেনেকে নহয় it is always B1 from B1 the electron will move to C from C it will go to C1 from C1 it will go to A and lastly it will come to A3 আৰু A3 so এখন হল তোমাৰ membrane আকিদি মই এতিয়া একিটা হল কমপ্লেক্স ৰাইট কেটা 1 2 3 4 5 ফিফথ কমপ্লেক্সটো তো ইলেক্ট্ৰনৰ নহয় সো ইলেক্ট্ৰনটো ইয়াৰ পৰা ইয়ালে আহিব ন লাষ্টত কি থাকিব সাইটোক্ৰোম A3 কি লব ইলেক্ট্ৰনটো লব ইলেক্ট্ৰনটো লৈ পেলাই হি কি কৰিব ম্যাট্ৰিক্সত ঘুমাই দিব ম্যাট্ৰিক্সত কি আছে অক্সিজেনে কি কৰিব ইলেক্ট্ৰনটো লব সো ইলেক্ট্ৰনটো ললেইটো নহব তাকটো প্ৰটনো লাগিব গ্ৰাজুৱেলি পিছত প্ৰটন আহিব সো হি প্ৰটনো লৈ লব আৰু তাৰ পিছত কি ফৰ্ম কৰিব water form কৰিব right so question ahe oxygen denote ki donate kune kore it is always cytochrome A3 that donates the electron to oxygen and where exactly is water form water is formed inside the matrix of mitochondria cytochrome A আৰু cytochrome A3 beleg নাম একেলগে দি থৈ দিছে কিন্তু একেলগে নিদিলেও হয় cytochrome A3 মানে একেলগে কি হৈছে just showing the transfer A পৰা A3 লে যাব হা It's B. Is it over? Eight upor question ahe. ETS by ETC, electron transport system by electron transport chain. Prokaryotes of kot thake, eukaryotes kot thake. Eukaryotes of to ami gome palu inner mitochondrial membrane thakibo. Prokaryotes to obviously mitochondria absent, so kot thakibo. Plasma membrane not kio specifically answer ki diba? that word where respiration takes place. Yes, so. So components kita likhiso, eta ami complex kitar kotha likhim, kuntu complex ot kuntu component thake. Next chart ekhon likha. FMN কি বস্তু
আগত কম্পোনেন্ট
FMN লিখিছো V লিখিছা flavin mononucleotide FMN full form What is the sound? Borokhon. Carbon monoxide, CO. So we are talking about the ETS, right, or the ETC, eketai bostu kot ase, inner mitochondrial membrane ot ase, ki ase, pasta complex ase, and within the complex there are certain components, and what are the components in each and every complex this chart talks about, right. So jedi precisely ba besi okoman tactfully question, if the question is asked, tethale question ahibo pare je eta complex ot ki component ase, question ahibo pare second complex or component ki, right, but first complex or component key. So it's advisable that you should remember the chart, right.
Second complex of succinate blade
Kio thake? FADH2 electron carrier. Tumi Krebs cycle ot sabason FADH2 code produce hoise aru enzyme kuntu associated hoy ase, succinic dehydrogenase, succinic acid, right. Krebs cycle or enzyme for the production of FADH2, succinic dehydrogenase, right. And this complex itself talks about that it will accept the electrons only of FADH2 and not of NADH2, right. And last class I already koisu eniki why the electrons of NADH2 enter the first complex and why can't the electrons from FADH2 enter the first complex, right. NADH2 electrons to iyat ahibo karon tat synaki enzyme to ase, right. FADH2 iyat jabo nware karon synaki nohoy, so kot jabo yare enzyme to second complex ote ase, to tahator cycle to ko prarambha hobo second complex, second over third, third over fourth, right. Okay, now up till here explanation has been done. Coenzyme Q is also called as ubiquinine, moi di dim baru, and the enzyme is this one. Okay, we come to this part. We have certain inhibitors of respiration. Jenika ami transpiration pohule, anti-transparent pohisu, inhibitors of transpiration, right. So kisuman ami antioxin pohisu, anti-gibberellin pohisu, mane first year we'll be talking about those hormones, okay. So we have some chemicals that will always inhibit the process. Photosynthesis will inhibit kore, we have talked about that. So in case of respiration also, the respiration process might get inhibited by the addition of certain chemicals, right. And out of these chemicals, this is something that is very commonly known. Cyanide khale manhu mori jay. Cyanide khale manhu kiho karne more? Karon respiration bondo hoy jay. Respiration bondo hoy jawa mane ki? Respiration mane to glycolysis ho hoy. Respiration mane to Krebs cycle ho hoy. So exactly ki hoy? The electron movement to bondo kori diye. If you notice, cyanide kot ase so third complex of electron to A pra kole ahibo A3 pra kole jabo oxygen le. So A pathway to block kori dibo. Jedi block kori diye, obviously oxygen electrons napabo gute process to bondo hoy jabo. So ultimately we have certain inhibitors that act according to the complex. If you want to stop the respiration process in the first complex itself, tetihale cyanide dile nohobo gitu. First complex ote bondo koribo lagile rotinone dibo lagibo. Specifically if you want from experiment point of view, no, kiteba to hobo pare that we want a respiration process where the electron movements get stopped in the first complex itself. So you need to give some certain types of chemicals over there. So complex wise chemicals were different. So third complex ot kune kam koribo? Cyanide. Cyanide into first complex ot kam no kore. Understood? So each and every complex will have a certain chemical, second complex or kunu nai. Understood? So out of this question kiya hai? Identify the respiratory inhibitors. Respiratory inhibitors, eto to obviously thakibo tar bhitorte, carbon monoxide thakibo pare, rotinone thakibo pare, right? Externally dibo. Internally, no, cell bhitor thakile to respiration e nohobo no. So cyanide kita khaba jetia finally aru, hoy na, so like you will have it externally itself, right? Or maybe you want to do certain experiments where you want to stop the respiration, then you will apply that cyanide. Clear? Clear? I mean, negative goes to class we should not be discussing. It's all for the experimental purpose that we are doing this. Okay. It is certain points about the chart.
associated with dicarbonate.
দিম মই ৰাখিব বাপৰে বিৰাট ইম্পৰ্টেণ্ট এইটো আহিব পঢ়িব লাগিব hydrogen is sorry. So details of this one, imankhini details of question aha nai. Question ki ahise, identify the mobile carrier of the ETS. So mobile carrier boli kole ta mane eta complex or pra antule ki carry kore, electrons bilak carry kore aru he karone tak ami ki boli kom mobile carriers. So kun bilak mobile carriers, coenzyme Q or cytochrome C are considered as mobile carriers. Bas that's all. Core pra kole carry kore iman question aha nai ha. UBQ or coenzyme Q eketai bostu. First point hoise niki Huh. Flavin mononucleotide. Write down this point. If this is over, please say. We will go to point number 4. While you are writing, I think you are understanding that line or the sentence also. Point 2 কি আছিলে? Electron carrier. Hydrogen is transferred from NADH2. NADH2 ক'ত হৈছিলে? মেট্ৰিক্সত. NADH2 ভাঙি গ'ল পেলাই কি ফৰ্ম কৰিলে? ইলেক্ট্ৰনছ, প্ৰটনছ আৰু NAD, ৰাইট? NAD কিয় ফৰ্ম কৰিলে? NADH2ৰ পৰা NAD ফৰ্ম কৰিব লাগিলে তাৰ হাইড্ৰজেনটো গুচি যাব লাগিব নালাগে? চ' সেই বস্তুটোৱে কৈছে। ৰিড দ্য লাইন। NADH2য়ে হাইড্ৰজেনটো ডোনেট কৰি পেলাই সি কিহলে কনভাৰ্ট হৈ গ'ল? NAD, ৰাইট? আকৌ NADয়ে কি কৰিব? হাইড্ৰজেন লৈ পেলাই NADH2 ফৰ্ম কৰিব। দিছ প্ৰচেছ উইল কিপ অন গোয়িং। অঁ, NADH2 ক'ত আছিলে? মেট্ৰিক্সত। NADH2 ভাঙি গ'ল, ইলেক্ট্ৰনছ, প্ৰটনছ দুইটা টিমত আহি যাব। টিমত আহি পেলাই ক'লে আহিব? FMN. চ' FMNতো ইলেক্ট্ৰনছো আহিব, প্ৰটনছো আহিব, খালি প্ৰটনছকেইটাক পুছ আউট কৰি দিব, ৰাইট? আৰু আফটাৰ ডোনেটিং দ্য ইলেক্ট্ৰনছ এণ্ড প্ৰটনছ NADH2 কিহলে কনভাৰ্ট হৈ গ'ল? NADলে, ৰাইট? চ' একচুৱেলি NADH2য়ে NAD কনভাৰ্ট কৰিব লাগিলে কি ডোনেট কৰিব লাগিব? প্ৰটনছ। পাছত, ৰাইট? অভাৰ। Okay, online students likha hol niki etu? Two electrons. Whenever it comes out from the matrix, either it's NADH2 or it's FADH2, the team member comprises of 2 electrons and 2 protons itself. It does not matter either we are talking about NADH2 or FADH2. The team always comprises of this, right? Why 3 ATP? It's not because of this. It's because of the pairs of protons that is pushed out from the electron chain. নহয় NADH2 পৰা দুটা proton ওলাব কিন্তু আমাক কেইটা লাগে তিনটা pair কাৰণ এটা pair responsible for the formation of 1 ATP. So if 3 ATPs need to get synthesized from 1 NADH2 আমাক কেইটা লাগিব ছটা না লাগিব জানো so ছটা কেনেকৈ দিব তাটা দুটাহে আছে কিন্তু that electron will move from one complex to the other complex and in the process তাহাতে oxidized reduced হৈ থাকিব so reduced হলে will show the pathway then you will understand right from these 3 lines খালি এটাই মনত ৰাখিবা 1 NADH2 3 ATP 1 FADH2 2 ATP আৰু the team comprise of 2 electrons 2 protons bas point number 4 likha cyanide sensitive respiration okay if I use two words cyanide sensitive or cyanide resistant cyanide sensitive respiration cyanide resistant respiration duita meaning buzi pao cyanide sensitive hole ki hobo cyanide dile mori jabo cyanide resistant hole ki hobo cyanide dileo no more ফিফথ পইণ্টটো কৰিব পাৰা हाँ हाँ बेंजाइल monochlorobenzyl hydroxamic acid. Okay, what does point number 4 and point number 5 talks about is, we have one type of a respiration that is most common in organisms, but living organisms, and that is cyanide sensitive, right? Cyanide delay respiration bond hoise jabo and as a result the organism will die, right? But we have something called as a cyanide resistant. So cyanide resistant example ami duta disu, cyanide resistant respiration, if you want to stop it, then we have to give certain types of inhibitors again. So tahator inhibitors or naam ki M-clam aru SHEM. Normal respiration or inhibitor buli kole, the most common inhibitor ki hoy cyanide ba carbon monoxide heblak hol inhibitors. E jar alternate system eta ase, tahatok jane inhibit koribo lage belek chemicals lagibo. So it is M-clam aru SHEM, right? Say. কি সেনসিটিভ সাইনাইড সেনসিটিভ টু নহয় এইটো সেনসিটিভ হৈ যাব ৰেজিষ্টেণ্ট ৰেস্পিৰেশ্যন বুলিতো নাই ড্যাশ ৰেজিষ্টেণ্ট কি হ'লে ৰেজিষ্টেণ্ট সাইনাইড হ'লে ৰেজিষ্টেণ্ট সো সাইনাইড দিলে নমৰে কিন্তু এই দুটা দিলে মৰি যাব ৰাইট এই দুটাৰ ক্ষেত্ৰত হা or maybe there are certain examples other than these two also right just you have to know that there is something called as cyanide resistant respiration also right okay ek blur moi ki koribo lage okay 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 eta ahise niki বেছি ব্লাৰ হৈছে এতিয়া আহিছে নেকি মই এইটো পইণ্টতে থাকি যাওঁ অ'কে আকৌ বেয়া গ'ল মই আকৌ পজিশ্যনত আহি যাওঁ এতিয়া দেখা পাইছা নাই okay tame moi yate thakibole center of attraction neki okay right once you are done please say full form gita di diso full form gita swa full form or upor questions ketia aha nai de it's always in the short abbreviated form the question might be asked right? You don't have to by heart this long names here monochlorobenzyl hydroxamic acid benzyl benzyl huh salicylic hydroxamic acid SALICYHYDROXYAMIC আগৰটো নিচিনাই right Is it done? Online students, is it done? Okay. Okay, now we'll draw the diagram that is given in your NCERT book. You can draw the diagram that is from the NCERT book, or you can draw the diagram that I give you. It's almost the same, right? NCERT book page number 2? Okay, please take out page number 233, those students who have the book with you. So, should I draw the diagram or you'll draw it yourself? Just go through. Do you all of you have your book? Okay, forget. I'll draw it. তোমালোকে নিজৰ নিজৰ বুকখন চোৱা ঠিক আছে হা লাগিব ড্ৰ কৰা হা ইয়াতে পাঁচটা কমপ্লেক্স নাই ইয়াতে পাঁচটা কমপ্লেক্স নাই কাৰণ এইটো ইটি আছে It is not oxidative phosphorylation. Oxidative phosphorylation is related with fifth. So, tathe ami fifth complex to akim. Huh? Oh, ekoi nai no aru. মোৰ মতেতো হ'ব লাগে মোৰ মতেতো হ'ব লাগে কাৰণ নোটছত হা that cannot be an excuse my dear NCERT বুকখন পঢ়িবই লাগিব কিহৰ কাৰণে পঢ়িব লাগিব মোৰ নোটছখনতো from understanding point of view it's very good but in the exams direct sentences from the NCERT book would be given মইতো ডাইৰেক্ট ৰিপিট কৰি লিখি দিয়া নাই ন মই বুজাইছোহে বস্তুটো হয় নহয় কিন্তু বুজোৱাৰ ওপৰত কুৱেশ্যনটো নাহে ন চেণ্টেন্সটো তাৰ পৰা আহিব চ' NCERT বুকটো পঢ়িবই লাগিব হয় This is one diagram from the point of explanation and understanding, right? If you want to refer to this correct drawing, you have to refer to the NCERT book. ঠিক আছে তাতহে বেছি প্ৰিচাইছলি দিয়া আছে মই যিটো ডায়াগ্ৰাম আঁকিম জাষ্ট বুজি পাবলৈ cytochrome C ৰ পিছত cytochrome C1 cytochrome C1 ৰ পিছত এটাই কমপ্লেক্স দেখাইছো A আৰু A3 লাষ্টত কোনে পায় A3 এ পায় The blue line indicates the electron movement, the blue and the red combination line indicates the proton movement. So, in the diagram what I am showing is just the electrons and protons from NADH2, FADH2 ro hobo, kintu moi dekhaisu ki diagram ot NADH2, NADH2 e first complex ot jai, first complex ot ki asile sosun component, just refer to the chart that we have drawn just now, FMN, to electrons kune lobo FMN, right? Next complex ot ki ase FES, come to FES, right? FES e ki koribo, it will transfer, right? So, yato present, FES e hodai electron to kak dibo, ubiquinol. So, in this case also it has given here, in this case also it has given here. Now, once ubiquinol takes in the electrons, it becomes excited. So, what it does is, it takes up the protons, gets reduced, pushes the protons, transfer the electrons, right? Moi je koisilo eta NADH2 within the journey 3 pairs of protons push out kore, right? 3 pairs of first pair to yar prai ahi gol, right? Matrix ot to okol eta NADH2 nathake no, there are so many protons and due to various reasons protons are found, right? Kiba kibi bhangile protons form koribo. So, accumulation of protons ase. So, next UQ jetia UQH2 le reduced hoi jabo tetia hi aru eta koribo. Ako UQ ase, UQ has to get reduced. So, he ki koribo hydrogen loi pelai push out koribo. So, ultimately how many pairs within the journey? 3 pairs, that will result in the formation of 3 ATP. UQA loi pelai UQH2 form kore. Hekane mo direct yat ne dekhi pelai ya. Either you can draw it here also. Confusion hoise jodi UKH2 etu eneke di dia. Proton to actually diagram basis ami yate indicate koribo lage, karon actually protons to kune loise UKH2 e ultimately loise no. Aru etu proton korpra ahile ami to yar proton to aka nai no yate etu korpra ahile karon yar pisot aru eta complex ase no. So, in that complex what they will do, they will push out the protons from the perimitochondrial space into the matrix again, and then from the matrix the oxygen will take in and ultimately get converted to water. So, this is all about the first step of terminal oxidation, that is ETS. Next, if instead of NADH2, if I draw FADH2, tat ki hobo electrons protons FAD, right? Tahate electrons protons FMN ok dibo janu, kod dibo UQ. So, ekhini process journey to arambo hoy, tetale etu pair to keteo liberate no hoy. So, keta pair liberate hobo 2, so 2 ATP. So, if this is over, oxidative phosphorylation heading koriba. Online students, is the diagram clear? Okay. Okay. If we are done with this, oxidative phosphorylation heading koriba. What is oxidative phosphorylation? The process of forming ATP. It is the second step of terminal oxidation. FES protein to thakiboi. First complex of ki likhisu sosun FMN. Second complex of ki likhisu coenzyme Q. FES to carrier. He proton to thakiboi tate, right? Iron sulfur protein to thakiye jay tate. Can I rub the board? Over now. So, in the diagram what we have seen, accumulation of proton in which area? Perimitochondrial space, right? I am not going to draw the diagram again. So, perimitochondrial space on NK accumulate hole ki hobo? Proton gradient. Proton gradient karone ki hobo? Proton motive force. Proton motive force or karone ki hobo? The protons will be pushed back. While they are pushed back, they will come through which pathway? FO-F1 particle, or in this regard we say complex 5. Right? Good. Five complexere humai jabo. Ulai tahate jikonu complexere ulabo pare. Kintu humuar homoy tahate fifth complexere humabo paribo. Right? Okay, oxidative phosphorylation likha. Okay, before starting this, are the two words clear? Oxidative phosphorylation or photophosphorylation? Ekta word to clear, doubt na eko, right? One related with photosynthesis, process of preparation of ATP, and this related with respiration, the process of preparation of ATP or forming of ATP. The second diagram is just to remember that what is the meaning of coupling factor? Nothing but the same thing, FOF1, right. So, the fifth complex FOF1 particle, coupling factor or proton canal, anything, right. So, coupling factor to dekha, it has a base, it has a stalk, it has a headpiece. একেটাই বস্তু, right? ATPase ক'ত থাকিব হেডত, হেড টু মেট্ৰিক্স, হেড টু ক'লে চাইডে থাকিব মেট্ৰিক্স, হা অঁ হা হা এই গোটেইটো মানে অকল সেইখিনি নহয়, অকল গ্ৰেনিউলছবিলাক নহয়, গোটেইটোকে আমি যিহেতু প্ৰটোন পাছ কৰিব ন, চ' বেছ পাৰ্ট এইটো ডায়েগ্ৰামত ক্লিয়াৰ হৈছে চোৱাচোন, এইখিনিক আমি কি বুলি ক'ম বেছ, right. So, it can be talked about it as coupling factor, it can be talked about as a proton channel, right. Question ahe, head to jodi matrixot ase, base to kot ase, inner mitochondrial membrane. So, this is important, base kot ase, inner mitochondrial membrane. So, fifth complex, so obviously if it's a fifth complex bilak kot thake inner mitochondrial membrane. So, fifth complex kot thakibo inner mitochondrial membrane, but a part of the fifth complex protrudes towards the matrix of mitochondria, right. Diagram over? Nai hua? Okay, akilo. একে এইটো পাৰ্টটো হৈছে চাগে So, oxidative phosphorylation, তাৰ পিছৰ ইনফৰমেশ্যনখিনিও লাগিব নেকি? এইখিনিতো আমি কেমিঅস্মটিক হাইপথেচিছত অলৰেডি লিখিছোঁ, right? So it's the same explanation, no point of writing it again. Proton gradient would be formed, proton motive force কি হ'ব? Proton canal through push up. তাৰপিছত কি হ'ব? Once the proton reaches the head piece, the enzyme gets synthesized, and because of the enzyme, ATP gets formed. Right? Where does ATP forms in a mitochondria? In the matrix of mitochondria. Where is water produced? In the matrix of mitochondria. Where is the enzyme present? Or where is ATPase present? Fifth complex, answer correct. Proton canal, answer correct. Right? FOF1 particle, correct. Head part of FOF1 particle, correct. Right? Huh? Oh, inner membrane. So ultimately we have completed the 3 steps of aerobic respiration, glycolysis, Krebs cycle and terminal oxidation. Is the last point understood? Coupling factor, we have already drawn what is a coupling factor, we understand, right? In case of terminal oxidation, both the process goes hand in hand. It's not like this, the electron movement will take place, it will wait for some time and then only the proton will go. Together, and so we have used a word called as coupled together. Right? Duita process ekeleke jage, kintu kisuman chemicals use korile ki hobo he process kitak stop kori dibo, and those are called as what? Uncouplers. You will inhibit the process. I think this is all for today. For the next class, eta heading kori thoi dia, bioenergetics of respiration. Bioenergetics of respiration. bioenergetics of respiration we will do in the next class. Together with that, Okkoman part baki ase, I think by next class we will finish this topic. What we are going to discuss, we are going to talk about this one. Second is we are going, I think you remember we have talked about an alternate process of glucose breakdown, right, pentose phosphate pathway or PPP, and we will be talking about RQ, right, respiratory coefficient. We have talked about the word called as RS. RS ki asile respiratory substrate. Eti ami ki porim respiratory quotient, right. So, ekini part baki ase, we will do it in the next class. So, what will study in this one? The total ATP that is produced, right, in each step. Ekini ami next class of porim. And that is all for today. Thank you.